Okay. Good morning. Apologies for being slightly late. I'll blame it on Metro today. Cynthia McOliver. I'm an environmental health scientist at EPA. I'm also a project officer, which means that I manage grants after they're funded. And I'm very excited to have an opportunity to talk to you about the previously funded food allergy, genetically engineered food. Several of the grantees have spoken this morning, and I'm just going to give you an overview of the 3 solicitations that funded these projects. First, I work at NCER, which is the National Center for Environmental Research. We coordinate the STAR grant program, which is the Science to Achieve Results program. We fund research grants, centers, also undergraduate and graduate fellowships. Many of you may have STAR graduate students in your academic institutions. And typically, our research is funded through RFAs derived from the STRAT plan, and then these RFAs are prepared in cooperation with other program officers within the agency. So we have writing teams that come together to develop the research idea, and then we have a kind of a very rigorous peer review process and then internal review process prior to funding. So EPA has supported a number of biotechnology research grants, and the link to these are available here, and I also believe that these slides will be available to you after the conference. And so we have funded three different solicitations. In 2006, we had the biotechnology potential allergenicity of genetically engineered foods RFA, all 4 of which are complete. And I'm not sure if you can read these, but I'll just try to give a synopsis of the areas of interest under each RFA, the methods proposed by the grantees, and then give like a brief overview of the results from 1 or 2 of the grantees. There's not really a chance to kind of walk through each of the several grants under each RFA. So under the 2006 RFA, the areas of interest were 1, to develop methods to assess the potential allergenicity of pesticidal proteins, testing of relative allergenic or sensitization potencies of proteins with and without a history of producing food allergy. And really the goal of the first RFA was to improve predictability of risk assessments for potential dietary allergenicity of exposure to novel pesticidal proteins in engineered foods. And as I mentioned, there were 4 grantees awarded. Dr. Vinny Ganger's lab, Dr. Vinny was here, and Dr. Hoganesh Haram, Dr. Rick Goodman, and Dr. Warner Brown. And for Dr. Ganger's project back in 2006 or 7, he really wanted to determine the sensitivity and specificity of mouse-based adjuvant-free transdermal food protein sensitizations. For Dr. Harm's lab, Hoganesh Haram's lab, they wanted to compare gene expression between to identify changes in gene expression specific for responses in food allergies. Dr. Goodman's lab, they looked at model protocols, and they tested these under native conditions to evaluate overall IgE binding, and also looked at cross-inhibition assays to demonstrate specificity of binding. Dr. Braun's lab looked at statistical ways to test validity of proteins in SDAP database, which you heard about today, using bioinformatics rules. And they also examined structure-activity relations among allergens by analyzing sequence requirements, RE, IgE epitopes. And just 2 general findings from these projects from Dr. Braun's lab. They identified new sequence similarity scores for linear epitopes, and they identified the use of property distance scores based on physical chemical profiles. Dr. Ganger's lab, there were kind of 3 major outcomes. They looked at allergenicity in the mouse model that they used were dependent on the number of exposures to the protein of interest. There were 7 human allergenic proteins produced, and produced a positive response in the mouse model, and they also identified amaranth seed protein as testing positive in the animal model readouts. The next RFA was in conjunction with the NIH-NIAID. And the title was Exploratory Investigations in Food Allergy. And all these projects, as well as the 2006 grants, have been completed. And the link on the bottom of the page, if you can see it here, is a link to the final reports as well as progress reports on these projects if you're interested in following up more specifically. And the main area of interest for these grants was to determine the mechanism of IgE-mediated food allergy and related comorbid conditions, using ex vivo studies with human specimens and studies with current or new animal models of food allergy. And as I discussed earlier, the middle section discusses some of the proposed approaches and methodologies in these projects. Dr. Barron's lab at Sinai used murine models of IgE-mediated food allergy to test the role of TSLP in the inductive and effective phases of food allergy. Dr. Goodman won again in this lottery of funding. I developed a standardized functional assay in the rat basophil cells that express human IgE receptors. They also tested legume tested legume allergenicity in an ex vivo IgE allergen-induced release assay. In Dr. Shine's lab, they used computational and experimental approaches to identify the structural and physicochemical properties of IgE binding sequences compared with clinical profiles. And finally, Dr. Sperling and Bryce's lab, they looked at novel murine models to assess the role of Fc gamma R3 signaling on dendritic cells in the induction of food allergy, and also looked at IgG-mediated processes on anaphylaxis. An example outcomes and findings from Dr. Shine's lab was that sequences from a major walnut allergen, JIGAR2, which were predicted to be similar to an immunodominant epitope of the peanut allergen, RH2, bound IgE in sera from patients who reacted to either walnut, peanut, or both. Secondly, the SDAP software accurately predicted the amino acid sequences that were similar between different nuts and of which of these amino acids was most likely to be recognized by IgE antibodies from the blood of allergic individuals. And finally, we come to the current round of grants. In 2009, approaches to assessing potential food allergy from GE plants. And the main areas of interest under this RFA was to address key gaps in an understanding of what makes a protein behave as a dietary allergen in order to improve risk assessment for pesticide proteins with unknown allergenicity. Secondly, we want to identify key unknown factors related to sensitization, oral tolerance, and elicitation of allergic symptoms that may enable predictions of whether food allergy would develop in individuals exposed to these proteins. And the main aim of this RFA was to improve safety assessment for GE plants. And there were, I'm going to discuss the three approaches for Dr. Ganger's lab, Dr. Braun's lab, and Dr. Barmert's lab. And luckily, these grantees are all here, and they've kind of given you an overview of their projects to date. Under Dr. Ganger's project, they looked at TSOE mouse model to determine NOAL and LOAL of plant-derived food proteins, and also exploring immune, clinical, and physiological responses to isolated pure food proteins. In Dr. Braun's lab, they expanded the SDAP web server by sequences and allergen-specific motifs of novel allergens, and also applying new prediction methods. Dr. Edkins' lab at Children's Hospital in Boston that tested F709 mice as a predictive probe of food protein allergenicity. And finally, Dr. Barmert's lab, they are conducting oral sensitization of C57BL6 mice, and they're measuring sensitization using immunoassay and peptide analyses. Some progress to date from Dr. Barmert's lab, they're doing method development for purification of peanut allergens. They're developing sensitized mouse models for allergenicity assessment of the proteins. They're using conventional mice that were orally sensitized and later orally challenged, did not have a significant drop in temperature. And finally, they're looking at clinical scores which are showing more variability than IP-sensitized conventional NGF mice. These grants are wrapping up within a year or two. The previous research data is available. As many of you know, some of these grants are very prolific in publications and presentations, and I'm very excited to have a chance for them to talk to you directly about their work. And I'm hoping in the next year or two to either develop an updated website with the grantee information, and maybe do a meta-analysis paper on the work produced under these grants. So if there are any questions for me about whether there's future funding on this topic, I can't really speak to that at this point, given current situation. But I think that this area of research is very important, and I'm happy that EPA had a role in funding such innovative work. My contact information is here. Give me a call or follow up with questions. And if you're interested in funding opportunities in the future, whether on this topic or others, our website is linked below, epa.gov/NCER/RFA. Thank you very much.